thank you very much for inviting me. It's great to be here, and I'm glad that I got here early because I got to go upstairs and see some of the robots, which are very cool. So if you haven't seen that, I recommend that strongly. Okay, so what I thought I would do today is give kind of a short talk, about 20 minutes, and just tell you our basic story. And then we can open up to questions, just so it's more informal and interactive. And I hope that you won't be shy, and even if you're in middle school, and will come to the microphone and ask something if it interests you. Okay, so let me start with a little bit of introduction. So as Mary Beth was saying, I'm a neuroscientist, and my research focuses on what's called neural coding. So what we work on is how the brain takes information from the outside world and encodes it into patterns of neural activity, which are electrical pulses. And then how it uses those patterns to allow you to do things, to see, to hear, to reach for an object. So I'm really very much of a basic scientist, and I'd never done anything clinically oriented before. But in the last 2 or 3 years, I started to switch over to take what we're learning about these patterns of activity and apply it to medical problems. And as she was saying, what we focus on specifically is developing prosthetic devices, new kinds of prosthetic devices. And what I wanted to do today is show you an example of this. So it's really our first venture into this. It's the development of a prosthetic device for treating blindness. Okay, so let me start with a little bit of background. So there are 2 million people in the US and many more worldwide who have advanced stage blindness. So blindness from diseases like macular degeneration or retinitis pigmentosa. And while there are treatments that are starting to be developed for the early stage cases, for patients with advanced stage disease, who are really at the end of the line, there's very little that can be done. So for these patients, their best hope right now is through prosthetic devices. The problem is that current prosthetics don't work that well. So they're still really very limited in the vision that they can provide. So for example, with these devices, patients can see things like bright lights and high contrast edges, but not very much more. So nothing close to normal vision has been possible. So just to give you a feel for what these devices are like, the way it generally works is that a patient gets an array of electrodes implanted into their eye, into their retina, and then current gets sent to each of the electrode tips. And so they'll see a spot of light at the tip of the electrode. So typically there'll be 64 electrodes, so 64 spots of light. So with this kind of thing, they can see where very bright things are. So they can walk into a room and see where a window is, because it's bright. They can see where a lamp is. And so they can strategically place the lamps around the room so they can, maneuver around and get to the, not trip over the coffee table and find their way around. But, it's nothing close to seeing faces or trees or, what's inside the refrigerator when you open the door. And so it's been a big limitation. And so what I'm going to tell you about today is a device that we're developing that I think has the potential to be much more effective. And what I wanted to do is just show you how it works. So let me start with how a normal retina works first, so you can see the problem that we were trying to solve. So here's a retina in the middle. So you have an image, a retina, and a brain. So when you look at something, do I have a pointer? Oh, I do, I guess, sorry. That's just the microphone, not the pointer, okay. I think I can do it with this. You have an image, a retina, and a brain. So when you look at something like this baby's face, it goes into your eye and it lands on your retina. On the front end cells here, the photoreceptors. So then what happens is the retinal circuitry, that's the part in the middle, it starts to process it. So what it does is it performs operations on it, it extracts information from it, and it converts that information into a code. And the code is in the form of these patterns of electrical pulses that get sent up to the brain. So the key thing really is that the image ultimately gets converted into a code. And when I say code, I do literally mean code. So like this pattern of pulses actually means that baby's face. So when the brain gets that pattern of pulses, it knows that what was out there was a baby's face. And if it got a different pattern of pulses, it would know that what was out there was, say a dog, or another pattern of pulses would be like a car. Anyway, you get the idea. Okay, and so in real life, though it's more complicated, it's all dynamic, meaning that it's changing all the time. So the pattern of pulses are changing all the time because the world you're looking at is changing all the time too. Okay, so it's a pretty complicated thing. Patterns of pulses coming out of your eye every millisecond, telling your brain what it is you're seeing. So I just showed you 4 here, but in your actual eye, in the human, it's 1 million patterns of pulses that are coming out. So about 10,000 in your central retina, but for your whole eye, it's 1 million. Okay, so what happens when a person gets a retinal degenerative disease? What happens is that the front end cells die. So the photoreceptors die. And then, over time, the cells and the circuits that are connected to them, they start to degenerate and die too. Until the only thing you have left, or really the only reliable thing you have left, are these cells here, the output cells, the ones that are sending the signals to the brain. But because of all this degeneration, they're not sending any signals anymore. They're not getting any input, so there are no signals to send. So the person's brain no longer gets any visual information. So he or she is blind. Okay, so a solution to the problem then would be to build a device that can mimic the actions of this front-end circuitry and drive these output cells. So they could go back to doing their normal job of sending the signals on up to the brain. So this is what we've been working on and what our prosthetic does. And so let me show you schematically how it's set up. So it has 2 parts, what we call the encoder and the transducer. So the encoder does just what I was saying. It mimics the actions of this front-end circuitry. That is, it takes images and it converts them into the Retinal code. And the transducer then sends the code on up to the brain. And you can have different kinds of transducers. So you can have electrodes, or you can have this new kind of transducer, which is called an optogenetic transducer. It's like basically a molecular transducer. And that's actually the one we use. And I'll tell you a little bit about that later and why we use that. Okay, and so with this system, this combination of the encoder and the transducer, you have a prosthetic system that can produce normal output. So a completely blind retina, even one with none of that front-end circuitry at all, can now send out normal signals the brain can understand. Okay, so this has never been done before. Okay, so I wanted to take a second and tell you about the encoder because it's really the key part. And it's the sort of, I don't know, cool and interesting part too. So what the encoder does is it's replacing the retina, really the circuitry of the retina, with a set of equations that we implemented on a chip. So it's just math. In other words, we didn't really make a device that mimics, with little mini devices that mimics each of these different cell types. We just abstracted what the whole retina does with a set of equations. So the equations act sort of like an encoder or a code book. Images come in, they pass through the set of equations, and out come streams of electrical pulses, just like a normal retina would produce. Okay, so now let me put my money where my mouth is and show you that this really is the case, that we really can produce normal output and what the implications are for making prosthetic devices. And before I forget, I just want to say we've only tested this so far in animals, not in humans yet, but we're getting there. Okay, so what I'm going to show you are 3 sets of firing patterns. The first one is from a normal retina. So what we did was we took movies of everyday things. We went to Central Park and we took movies of children playing and people walking and trees and squirrels. And we presented these movies to a normal retina and we recorded the firing patterns. Okay, so these are the firing patterns from a normal retina that was viewing these movies. And you can see it's, kind of complicated patterns. So then we took the same movies and we presented them to a blind retina under 2 conditions. So in one case, we presented to the blind retina doing it through the standard prosthetic, and in the other case, we presented it to the blind retina using our encoder transducer prosthetic. So let me show you the standard one first. Okay, so these are the firing patterns produced by a blind retina viewing movies through the standard prosthetic. And you can see that with a standard device, the cells will fire, but they're not firing normally because they don't have the code incorporated, and so it's sort of a smeared out kind of firing pattern. Okay, what about with the encoder transducer method? You can see that it's much different. The firing patterns very closely match the normal. So just to emphasize it, so what you're looking at here is a completely blind retina that has no photoreceptors at all, but now it's firing normal. Okay, so we have the code. We can produce normal firing patterns. How important is this? You know, what's the potential impact on a patient's ability to see? So I'm just going to show you one experiment, one sort of bottom line experiment, and it's called a reconstruction experiment. And so what we did was we took a moment in time, you know, just some moment in time from one of these movies, and we said, can we reconstruct what the retina was seeing at that moment in time? Can we reconstruct what the retina was seeing from these firing patterns? So we did it with the standard prosthetic, and then we did it with the encoder transducer one. So let me show you with the standard one first. Okay, so you can see here that the firing patterns, that the reconstructed image is pretty limited. So the image produced by the standard prosthetic doesn't really show you that much, and it's because the firing patterns aren't normal, so they just don't carry that much information. You can sort of tell that there's something there, but you can't quite tell what it is. Here's what it was. That baby's face. Okay, so what about with the encoder transducer? And you can see that it's much better. So not only can you tell that it's a baby's face, but you can tell that it's that baby's face, which is a really challenging task. So on the left is the image produced just by the encoder device itself, just to show that we really do have the code. And the one on the right is actually from real retinal tissue. So you lose a little bit of information as you go from the encoder to the transducer into the actual tissue, but it's not too bad. Okay, so I just wanted to take a second to say something about the standard prosthetic before I go on. So when the standard method first came out, and this is like 15 or 20 years ago, it was really an exciting thing, you know, just to be able to make a blind retina see at all. And it didn't work that well, but nobody was worried about it really, because the assumption was it's just a resolution problem. And if we just, you know, so meaning that if you put more stimulators in, then you would just be able to boost it up. But it's not true. It's not true from first principles, and it's not true as I'm showing you empirically here. So here we stimulated and recorded from 10,000 cells. So as many cells as would cover your whole central vision. And you can see that that it just doesn't get good enough, really. So what I'm saying is that the standard method has an inherent ceiling on it. It can get better up to a certain point, and then it gets stuck. But if you add the code, then you can break through that ceiling, maybe all the way up to normal vision. And so this is our contribution, figuring out the code and building it into a device. Okay, so what does it look like, or what is it going to look like as we get it into patients? So I was just wanted to show you this. Not sure how well this shows up on the screen, but a key aspect of the approach is that it doesn't involve any surgery, just an injection into the patient's eye, which can be done in a doctor's office. So that device that I showed you before, it doesn't actually get implanted into the eye. It's just embedded into a pair of glasses. And so what we do is we inject a compound into the eye, and then the device then interacts with it. So I'll just show you. So I gave a TED Talk on this once, and so when you give a TED Talk, they give you a graphics designer who makes all your slides for you, and that's why my slides are so nice, because I would never be able to make slides this good. But just so you know, the glasses don't really look like this. They're actually like ugly ski goggles, but the graphics designer wanted it to look like this, but I wanted to be honest and confess to you. But the key thing is that because all of the electronics are in glasses, it means that we can upgrade. So we can make them look nicer. We can make it. Add components to improve the resolution, or to improve, add color vision. Actually, we already have a lot of color vision built into it, but we don't have to make the patient suffer while we're doing that. Once they have that injection, they're done, and everything else is external. And that makes a huge difference for improving things and allowing us to move forward quickly without having to have everything perfect before we start. Okay, so since I started doing this, I started a company to raise money so I could develop this, and I just raised the money about 10 months ago. And I just want to give you like a quick update on what we've been doing since then. And I'll just run through it in 2 seconds. So we've incorporated the code for primate retina. So what I showed you was mouse. So we've now got the code for primate retina, which means includes humans. And so it's now appropriate for use in humans. We minimize the electronics in the device, so it's now small enough to fit into a pair of eyeglasses, well, goggles, as I showed you. We increase the number of cells the device can stimulate, 10,000 cells, so we can now target the entire central retina for vision restoration. We tested 9 vectors for delivering the gene and identified 2 that are suitable for the clinical trial. We performed the first round of safety studies, the device alone, and currently running the second one. And I put together a team to perform the clinical trial. And so I'm not going to go through the whole timeline, but basically we should be able to get our way through the FDA by the end of the year, and then we'll be able to start our first clinical trial. Okay, so before I wrap up, I just wanted to say that that the strategy that we're using generalizes. So the same mathematical formalism that we use to figure out the code for the retina can also be used to figure out the code in the auditory system for hearing and in the motor system and other places as well. So in principle, just the way we were able to jump over the damaged retina to get to the retina's output cells, we could jump over the damaged cochlea, the hair cells in your inner ear, to get to the auditory nerve, or jump over parts of your motor system to bridge a gap produced by a stroke. And I think I'm just going to end with one sort of a simple message, which is understanding the code really matters. So if we can understand the code, the language of the brain, then things become possible that didn't seem possible before. Thank you, and I just want to thank the people that helped. In particular, the first guy, Chase and Tanner. Thank you.
So thank you, Sheila. So as Dr. Nirenberg said, she's really open to answering your questions. So I'd like to encourage not only adults to ask questions, we'd love to hear some kid questions as well. And you can ask about the science, but you can also ask if you're curious about maybe how Dr. Nirenberg got started, and maybe sort of what educational path she might have followed, things like that. So definitely don't be shy about asking questions.
What do you like the most about your job?
Sorry, what did you say?
What do you like the most about your job?
I think that the thing that I like the most is it's completely absorbing. I'm solving a problem and I'm not watching the clock, I'm not waiting for a lunch break, it's just totally interesting. What's figuring out the code of the brain and all the implications for it. And we also make robots, and so figuring out the implications of using the code to direct a whole other path of research.
How long did you have to go to school to become a scientist?
Well, you have to get a PhD. So I took a long time to do it. So, it's between 5 and 7 years or so to do that. But while you're doing it, you're still doing science. It's not like the graduate program you're on the way. But the upsides are, you're always in school. I always wear jeans. I never have to dress up. So it's just a prolonged adolescence forever.
How did you get interested in your job?
Where's the person who's asking that? I can't see where you are. Oh. Well, the brain is just a fundamentally interesting thing. It's what makes you, you, and me. I honestly can't think of anything more interesting than understanding human nature. So I was always driven that way.
What's injected into the eye, and where does it get injected?
What's injected is, it's basically a combination of gene therapy and a device. So what's injected into the eye is a gene that expresses a transducer. So it's a light-sensitive protein. We send a signal that activates it, and it causes the neurons to send electrical pulses. So it's just like it's a virus, actually. So this is how gene therapy works. It's made of a virus, a safe virus, it's like a cold virus. And it just goes one injection into your eye, and it integrates into the cells that are in your retina.
Disease that makes you go blind? What's it like? Like, what do you see?
Well, as you start to get these, there are many different kinds of blindnesses, but the ones that we work on are degenerative diseases. So what that means is you started out normal, and then over time, as you got older, the image in front of you started to get more and more blurry and darker and sort of kind of gray out. So some people can see in their peripheral vision, so they can still walk around, but they can't see faces. So if you ever are interested, you can look on the web, and they'll show you what the world looks like to a blind person, so you can get a feel for it. It makes you grateful that if you can see, to really appreciate it, because it's so hard to be blind, as you can imagine.
When you, the prosthetic, can it see patterns and colors?
I'm sorry, when we make the prosthetic, can they see?
Well, when you do the injection into the eye and they wear the glasses, can it detect colors and patterns?
So we haven't done it in people yet, but in principle, that's right. We're making the retina behave just like a normal retina. So the signals will go to the brain, and if the brain will be able to tell you what the patterns are, what the colors are. The reason I showed you the pictures in black and white is because we started with a mouse, and mice don't really see much in the way of color, so we just did the easy version for the pilot experiments, for the first experiments.
Could you apply this technology to like dreams and memories and like producing images from them?
What was the second part of it for?
And produce images from them?
I don't even know where you're. Can you raise your hand and so I can see where you are? Okay. What'd you say, if I can apply it to dreams and memories?
Yeah. Could you apply the technology to that?
Well, someday. I mean, so the dream isn't so much happening in your retina, it's happening in your brain. So there's a lot of money being invested in trying to make brain-machine interfaces so that you could stimulate the brain and experience things. So even if I'm not personally doing it, this is going to happen sometime soon.
What if you got blind?
Well, I worry about this sometimes because I play with dangerous equipment in my lab. But needless to say, if I got blind, it would be hard. I guess the people in my lab would try to keep doing it anyway and try to help people.
When you try it on humans, if it works, do you think you'll ever get it into contacts instead of glasses?
I would love to do that. So that would be the much better way. And I'm sure that every blind person would rather I did it with contacts than with glasses. But the way it works really is you do the first version of it in your clinical trial, and then you just do it with the goggles just to make sure everything's going to work and to work out all the details of it. Once it works, then a lot of rich people will put in money, and they'll help me. They're rich blind people, and they'll help me make it the fancy version with contacts. But first, we've got to prove that we can really make blind people see.
If someone breaks their glasses, do they have to go to their doctor, or can they just pick it up at like CVS pharmacy?
CVS pharmacy, I think that will work, or an optometrist. But they don't need anything fancy like this.
How did you know how to write the equations for you called it the transmitter thing? Transducer.
The encoder, yeah.
The encoder, sorry. How did you know how to write them? Because you said there were equations.
Right, so you have to study some, learn some math. So I want to encourage you because, especially since, most of you are so young, and you sit in math class and you wonder, like, why am I learning this? What is it? How does it help anybody? This is how it helps people. The time delay between what you learn in 8th grade and when you actually use it may be long, but you could do it faster than I did. But you have to learn enough math. This was algebra, basically, linear algebra, and probability. Those are two of the biggest ones to learn.
What's the process for collecting the data in the retinas that you guys used?
So what we do is the thing that's convenient about a retina is that you don't have to record from it in the live animal. The retina doesn't get feedback from the brain. So it's like a little isolated thing. It's a little neural network that you can take the tissue out and put it in a dish on a bed of electrodes. So we can present it with movies and stuff and record the electrical activity right in our lab in a setup without having to put the animal under, anesthesia if we don't have to.
When you test this on animals, how do you tell if the animals are blind, and if they're not blind, how do you get animals that are?
So just like people, animals have these diseases too. And so they're animals that will get the same disease, and you can actually buy them from a company, animals with a retinal degenerative disease. I know that seems weird, but you can just order the mice to come to your lab. So I know, and then you can see that they're blind, and then you can make them see.
Is your job very stressful?
Yes, I work a lot, but it's fun. It's like the first person was asking me. I'm absorbed, so even though it's stressful, I'm not, I'm happy when I'm doing it. Then when I get home, I'm still stressed and I have to watch Netflix for a while before I can fall asleep. But it's like everybody else. We all have a lot of stress.
Do they need to be tuned at all, or does it just work right away? Like, would someone say, I want it to look better, or do you have to dial knobs or anything? Or, you hook it to a primate, it just sees.
Well, we haven't done it in humans, so the way it is, you test these things on animals as much as you can. And then at some point, there's no more predictive value to do it. You're not learning anything more, and you're ready to go into humans, as long as it's safe. And that's sort of where we are now. As long as we can be sure that we're not going to hurt anybody, then we can take it into a clinic, and we will give the person the goggles, and then they'll give us feedback. And they'll say, I can't, it's not centered, or I'm not seeing as much detail as I thought, and then we can tune it to what their needs are. So we test it as far as we can in animals, because of course the animal can't tell you anything, and it's very hard to assess it. And so then you reach a point where you just have to do it in humans to get the feedback. And I just want to say something about the patients that we would do this on. You pick smart people who are interested in this, and they become your collaborators. So they're like, even though they're not scientists, they're so interested, and it makes their disease meaningful to them, because then they can help others. So you become a team, you and the patients that you're testing it on. Together, you're making this thing better for everybody.
How do you know the animals are seeing anything?
So there are two ways. One is we can see that their firing patterns are normal, so we know that they're sending the same message to the brain that the normal retina would be sending. And then we also did it by having them track images. So we hold the mouse, and it's staring at a screen, a blind mouse, and we send images by, and then we convert the images into the neural code, and then suddenly they can see it and can track it. And we keep them happy, and we give them popcorn and those cheese peanut butter crackers to keep them, they got to eat something while they're at the movies watching our little thing.
How did you come up with the idea to make the thing?
How did I come up with the idea? Is that what?
Yeah.
I don't know. Is that what the question was?
Yeah, how did you come up with the idea?
Well, I was working on, I'm just interested in how the brain works, period. So I was working on something to try to understand how neurons communicate with each other. And then I was talking to a student in the lab, and we were talking about whether we could make this really work to help blind people. And we were thinking about doing it with electrodes, but electrodes are not so good. And then there was a realization, if we use this molecular transducer, suddenly we could actually make a device that that could restore sight to blind people. And then once I thought of it, then I had to do it because how could you walk away from that? So that's how it happened. It just sort of unfolded.
If something did go wrong and you tested it on humans, what's the worst that could happen?
Well, what I'm doing, the vector that I'm sending into the eye has already, I'm building it on a vector that's already existed. So I just took out the other gene and I put in our gene. So it's a vector that's been in eyes for 7 years in people, and nothing bad has happened, knock on wood. So odds are nothing will happen. And if it turns out that the whole thing didn't work, then the person just has this thing in their eye, but it won't bother them. It won't do anything. It'll just be, they won't even know that they have it. It only works if it interacts with the device. So if it turns out the device didn't work for some reason or it hurt them, they just put the device away and then they're just back to normal. And they're no worse off than they were when we started, is what I mean.
Hi, thank you for your talk. My question is, I wonder if there are any eye conditions which result in one of the two eyes having this disease or degeneration but not the other, so that you might be able to track signals going into the brain from an eye that's not damaged and compare those with the damaged eye that you're trying to work on with the prosthetic. And if there are conditions like that, is that part of your plan to roll out the device to test it in that scenario, or do you have any comments on that?
That's exactly what I would like to do. Maybe not quite as extreme as having it be a totally normal eye and a damaged eye. I think the FDA won't let me do that. But what I really want are cases where one eye is significantly better than the other. Not exactly for the reason you said, though. It's partly because if the person is too blind, then their synapses in their cortex, in their brain, are not so ready to receive the information. But if they can see out of one eye, their brain's still in good shape. And that means what we send in will be ready to be received. Because the two eyes send information to the same place. So if the one eye is doing it, the brain's still going and it's ready to get the other information. So we're looking for people who have a little bit of a discrepancy, and we would go after the bad eye. I'm hoping we can find that. So.
Why does your brain know what you're looking at?
This is how we evolved. It's like first you had jellyfish or something who just could only see light and dark. And then over time you build up all these creatures that can see better and better things. Like we can see faces and all of those details. It's why is a hard question to answer. It's how, really, it does it. Someday maybe you can come to my lab and I'll show you. That's the easier, otherwise it will take me a long time to explain.
Who and where are your team of scientists?
Well, I'm a professor at Cornell, so my team of scientists are the researchers that work in my lab with me. So they're called postdocs. They have PhDs, and we're all very committed to doing this together. And it's in New York City.
Have you ever blinded an animal to test your prototype?
No. So this is a big issue because I wanted to test this in monkeys, because if I tested it in monkeys, then I would feel I was testing it in an animal closer to human. The problem is that there are no blind monkeys, and you can't just. So I didn't want to blind a monkey to do it. But I'll tell you the two reasons why. So one is, see, with the mice, they're already blind, so you don't feel bad about it. But monkeys are also very smart. So even if I didn't completely blind them and knock out every single photoreceptor, they're smart and they would cheat. And I wouldn't never really know if they were seeing because I made them see or because they found a way to outsmart me, right? And see with it out of the corner of their eye. And the idea of doing an experiment where I blinded a monkey for a bad, sloppy science that I didn't believe, I just wouldn't do it. So I called the granting agency that gave me the money, and I said, I really can't do this. Can I use it for safety studies instead? And they let me use the money for the safety studies. So the answer is no, I didn't blind a monkey.
Do you have a website that like contains news on your research and what you're doing?
Yeah, so I have a website, and I explain all the research, but it's sort of for scientists, so it's a little bit hard. But there have been a lot of news articles and stuff, and if you Google me, you'll find versions of it. And there's like a BBC documentary and stuff. It's easier to follow.
Can you see it in the dark? The glasses.
Can I what? Can
Can you see the glasses? Can the person see the glasses in the dark or the animal?
I can't quite understand the question. Did somebody translate that one?
She was wondering, when you have the glasses, will you be able to see in the dark?
See in the dark. Oh, well, so that's a whole other angle of this. So when I started doing this, the military got interested in making soldiers that were like bionic soldiers, so they could see in the dark. And we could include sending in messages like ultraviolet light and infrared light. Infrared is what you'd use for the dark. So in principle, if this works for blind people, then we can also make bionic people. But my goal first is to help the people who need it before we make superhuman soldiers, one thing at a time. So
I think we're just going to take maybe 2 more questions real quick.
All right, so once this is fully developed, what would be the cost of having this transplant in the glasses?
What will be the cost?
Yeah.
I don't know the answer, and so I would like to do it as least, as inexpensively as I can, so that everybody can have it. Because it doesn't involve surgery, I don't think it has to be super expensive. It can be just an injection, and then the glasses are pretty cheap to make. People who invested in the company might not feel that way because they need to recoup the money that they invested. So we'll have a little negotiation to haggle that one out. But I'm on your side. We'll try to make it as cheap as we can.
What is the iris really used for?
The iris, it controls the size of your pupil and opens it and closes it to let more light in. And it's colored, that's the part, but it's not, the color part isn't so important. It's really for controlling the size of your pupil. You know what that means? Okay.